Hello all, welcome to another topic in microbiology. The topic is culture media. Culture media is an artificial environment resembling the natural conditions necessary for the growth of bacteria. So we are trying to provide an artificial environment for bacterial growth as culture media. Let's see the history of culture media. First culture media was introduced by Louis Pasteur, the father of microbiology, and he introduced liquid culture media like urine or meat broth. Solid culture media was introduced by Robert Koch. He is known as the father of bacteriology, and he initially used cooked cut potatoes. And another name to be remembered in the history of culture media is a lady scientist, Frau Hess. She suggested agar as a solidifying agent in culture media. So Louis Pasteur, liquid culture media, Robert Koch, solid media, and Frau Hess introduced agar as a solidifying agent. What are the basic requirements in a culture media? There should be source of energy, carbon and nitrogen. We prefer to have salts like sulfate, phosphates and chlorides and carbonates of sodium, potassium, magnesium and calcium. Trace elements like cobalt, manganese and molybdenum. A satisfactory pH, that is slightly alkaline pH 7.2 to 7.6, which is the most optimum pH for most of the pathogenic bacteria. There should be adequate oxidation reduction potential, and some bacteria require some special growth factors. For example, the bacteria Haemophilus requires growth factors like X and V factor. Don't be confused with blood clotting factor. X factor is actually hemin. Chemically it is hemin, and V factor is nicotinamide adenine dinucleotide, NAD. So these two factors are necessary for the growth of the bacteria Haemophilus. What is actually the characteristic features of an ideal culture media? An ideal media should give you a satisfactory growth from single inoculum. Inoculum means what we are inoculating to a culture media is known as inoculum. So from a single inoculum, you should get satisfactory growth, that too in a rapid way, rapid growth, easy to prepare, it should be economically cheap, and this media should be able to demonstrate all the characters of the bacteria which is grown in that media. So these are the characters which we demand from an. Let's see what are the common ingredients to prepare a culture media. There should be water, preferably distilled water. The next ingredient is peptone, which is a complex mixture of partially digested protein. This peptone can act as a source of nitrogen and at the same time can act as a buffer also. We want meat extract, which is actually hot water extraction of lean beef. It is available commercially as Lab Lemco. Blood, either sheep blood or horse blood, and we can also use human blood. Serum, yeast extract, which is an extraction of autolyzed, and the most important ingredient in solid media is agar. Agar is actually a solidifying agent in culture media, which is obtained from seaweeds, and the most important thing is it's not having any nutritive value. Agar melts at 98 degrees Celsius and sets at 42 degrees Celsius. It is obtained commercially as powders. There are two varieties, New Zealand agar and Japanese agar. And if you are going to prepare a solid media, the percentage of agar to be added is 2%. And for a semi-solid media, which is having a jelly-like consistency, Agar concentration is only 0.2 to 0.5. So these are the common culture media. About the distribution of culture media, media can be dispensed in test tubes, petri dish, or in screw-capped bottles. So either petri plate or test tubes or in screw-capped bottle, media can be taken. Culture media is classified based on various criteria. First one is based on its consistency, classified as solid media, semi-solid media, and liquid media. Based on constituents, culture media can be simple or complex. Based on the nature of constituents, media can be synthetic or defined media, semi-synthetic or semi-defined media, and special media. There are about 9 types of special media, which is denoted as enriched media, enrichment media, selective media, sugar media, storage media, differential media, indicator media, assay media, and transport media. A variety of 9 different types of special media are available. The fourth classification is based on oxygen requirement, aerobic media and anaerobic media. We'll start with solid culture media. It is used to study colony morphology of bacteria. Colony means a clone of cells originating from a single parent cell. So, when you cultivate bacteria in a solid culture media, the growth of bacteria is observed as colonies. Each bacteria is having typical colony morphology. So that can be easily studied by using a solid media. In solid media, you can study special characters of bacteria like pigmentation, that is colored end products, whether bacteria can produce or not. Then hemolysis, lysis of RBCs are studied in solid culture media. Identification of bacteria is more easy when you cultivate in a solid media, and you will get the bacteria in pure form. You can easily get pure colonies of bacteria in solid culture media. And the very common example is that you're observing in this picture, nutrient agar media, and you can also see bacterial colonies, bacterial growth in nutrient agar media. So these are colonies in nutrient agar media. Can get isolated single bacterial colonies can be obtained in a solid media. About a liquid culture media, it is used mainly to prepare bulk cultures. Bulk cultures means when you want a large quantity of growth of bacteria, usually for the production of antigens and vaccines, we prefer a liquid culture medium. For culturing very large inoculum, inoculum means anything that is inoculated to a medium. If the quantity is very large, it is not possible to cultivate in a petri plate. So then we can take a liquid media taken in a large flask or large bottle. So when the inoculum volume is very high, like the inoculum is blood or water, then a large volume is preferred for cultivation. So when the inoculum is expected is very large in volume, then you can take a liquid media. And the common example is peptone water. This is a liquid media taken in test tube, peptone water. In liquid media, growth can be identified by turbidity. Turbidity means a clear liquid media will become cloudy or turbid. Surface pellicle. Surface pellicle means a membrane-like growth can be observed on the top of the media. Surface pellicle. Sometimes there will be deposits, and according to the bacteria, sometimes if it is a pigment producing bacteria, the color of the media will change if it is a water soluble pigment, and Some bacteria will give you typical smell or odor. So that's how we are detecting growth in a liquid media. The disadvantage of liquid media is that, like solid media, you will not get individual colonies. So isolation and identification is not possible in a liquid media. So if you are going to cultivate in a liquid media, after getting growth in a liquid media, from this you have to subcultivate to a solid media. Then only you will get isolated colonies of bacteria. Third is semi-solid media. Semi-solid media is usually used for the storage of bacteria and also to study bacterial motility. In semi-solid media, the agar percentage is very less, only 0.2 to 0.5. So, uh, uh, because of that consistency, bacteria can move. That's why it is used to study bacterial motility. And the common examples are mannitol motility media and semi-solid nutrient agar media. The next category of media is simple media. As the name indicates, it is a basal media made of common ingredients. It is used for the growth of non-fastidious organism. Non-fastidious means bacteria which do not require any special growth requirements. Only the basic ingredients are enough for their growth. They are known as non-fastidious. The simple media is used for the growth of non-fastidious bacteria. Based on consistency, it can be solid or liquid, and the common examples are peptone water or peptone broth and nutrient agar. The term broth indicates liquid media. So peptone broth or peptone water and nutrient agar are examples for simple media. Complex media means it is used for the growth of fastidious bacteria, that is, bacteria which require special nutrients. So this media also contains something special. Common example is blood agar media, which contain blood as a special ingredient. The term synthetic or defined media indicates we know the exact chemical composition of the ingredients used in this media. Then we call it as a defined media. Example is citrate media. Synthetic or semi-defined means the exact chemical composition of the ingredients are not known. We are not sure about the chemical composition of ingredients. For example, peptone water. So that's about the culture media, the classification of culture media, and the rest of the classification, rest of the criteria of culture media we'll discuss in the next class. Thank you.